So this is in container week 10, the seminar, because we reverse the order between the male and female genital organs to follow the lecture's order. And we will start with the vagina number 75. This is a longitudinal section with hematoxaniosin staining. So this is an organ with a lumen, so it has a mucosa, which is incomplete because there is no muscularis mucosae in this mucosa. Generally, in the genital tract, there is no muscularis mucosae. So this is true in general. And at this low power magnification, what we can see is at least two things. One, that the epithelium is very pale. And second, that the wall generally looks pinkish rather than reddish. Which means, taking the staining, hematoxaniosin, that the majority of connective tissue fibers here is elastic fiber to allow expansion of the wall of this organ, which is needed because during delivery it becomes a birth canal, so the baby needs to go through the lumen, and this assumes a considerable elasticity. Now, regarding the epithelium, if we enlarge, we can see that this is a stratified squamous non-keratinizing epithelium. You can see squamous cells on the surface. And as we mentioned, these epithelial cells, especially in more superficial cell rows, are very, very pale, basically unstained. And this is characteristic for this type of epithelium, not just in the vagina, but elsewhere also. If you remember, the lining epithelium of the esophagus is the same type of epithelium. And we could also observe this phenomenon there. And generally the reason is that this type of epithelium synthesizes and accumulates lots of glycogen inside the epithelial cells, and we lose the glycogen during the formalin fixation. So that's a chemical artifact. And here in the vagina, this feature of the epithelium, that it produces lots of glycogen and it is released to the lumen, has at least two biological significance. One, that there is a natural bacteria flora which lives in the lumen of the vagina, and these bacteria live on this glycogen to a substantial extent. And why is this important biologically? So this is a symbiotic bacteria flora, which is naturally here. So the first significance that these bacteria produce lactic acid as a metabolic end product, which makes the lumen of the vagina kind of slightly acidic. And this is what is important from biological point of view because of two reasons. The first reason that this acidic environment gives a certain degree of protection against pathogens, which is needed because this organ has an open connection to the external world. And the second importance of this acidic environment is that sperm cells don't like the acidic environment, which means that this acidic environment creates a kind of selection among sperm cells, kind of first selection. So the less fit ones cannot go on, but die from this acidic environment. So that's why the glycogen production is important, because of these two reasons. Defense against harmful pathogens and selection among sperm cells. Also, we need to mention that this kind of epithelium, the certified squamous non-keratinizing epithelium, is the second most resistant type of epithelium of the human body in terms of mechanical stability. The only even more resistant is the keratinized version, which covers our skin. And this mechanical firmness is needed here because, as we mentioned, the newborn needs to go through this canal, the birth canal, and obviously this will cause a considerable friction over this surface. It's a mechanical stress. Well, 9 months earlier, there is another mechanical stress, if you understand what I mean, which this epithelium also suffers. So these two things at least makes it understandable why this epithelium needs to be resistant enough. And this mechanical resistance is further increased by the many and deep connective tissue papillae, what you can see here. You know, one of the functions of these structures is to increase mechanical stability by making the interface surface between the epithelium and the connective tissue wavier, which means an increase in this attachment surface. And if you have a bigger surface area for attachment, then obviously firmer attachment can be established between the neighboring tissues. Obviously there is a basement membrane in between, but we cannot see it because it is quite thin. May I ask your thoughts about the nature of the anchoring junction, which anchors cells of the stratum basale here to the underlying basement membrane? Hemidesmosomes and focal adhesions, yes, which are obviously visible only in the electron microscope, not here. Another thing, what it is not new either for you, hopefully, these structures look like connective tissue islands within the epithelium. What are these? Yeah, connective tissue papillae, and they are not really islands. They have a connection with the rest of the lamina propria, but we cannot see the connection within the plane of sectioning because it is curvy. So it's outside of the plane of sectioning, but these are just the very same kind of connective tissue papillae. Yeah. So, you know about the stratum basale, we have a rather thick stratum spinosum or polygonale, where the desmosomes interconnect cells, and the uppermost few very squamous sero altogether is called stratum planocellulare or stratum superficiale. These are the three sublayers in this kind of epithelium. This is repetition. Now, what else? So we enter the lamina propria, very pinkish, lots of elastic fibers, and because there is no muscularis mucosae, no submucosa either. So after the lamina propria, the external muscle layer comes immediately, which is rather thick, actually the thickest layer in this wall, entirely smooth muscle. And this muscle layer is divided into two sublayers, not very distinctly, but more or less. It is the descriptions usually say that there is an inner circular sublayer. Well, as you can see, it is again a little bit unclear. So definitely these are smooth muscle cells in cross section. So these are really arranged in a circular manner around the lumen, but if you look at this territory, I wouldn't say it is very definite circular arrangement, because here smooth muscle cells are rather in longitudinal view. But this is what descriptions usually say, that there is an inner circular and an outer longicular sublayer, which is more believable. This outer longicular sublayer is much better defined. So these are really longitudinally sectioned smooth muscle cells here. That's much clearer and much more obvious than the presence of this inner circular sublayer. And the outermost tissue layer is adventitia. There is no serous membrane coverage around this organ. And in the wall you can see peripheral nerves, like here. This is in the adventitia. Of course, there are blood vessels around also. And also within the muscle layer you can see peripheral nerves, like here. Here also quite big nerve fiber bundle here, also here. Please realize that the color of the nerve fiber bundle is even paler than that of the smooth muscle, which aids recognition. And one more important feature what we need to emphasize here, that here in this wall, the connective tissue and the muscle tissue is very much intermingling with quite large blood vessels, as you can see. This is a elongated longitudinal view of a vessel also. And because of this arrangement that we have a kind of intermingling smooth muscle and connective tissue with lots of blood vessels, this is very much resembling the structure of the corpora cavernosa of the penis. And the corpora cavernosa of the penis are the real erectile tissues. Those tissues what we call erectile. Well, this is usually called as semi-erectile tissue. It's not so erectile, but quite similar. So during sexual stimulation, these vessels also filled up with blood, causing kind of rigidity in this wall. This looks like a lymphatic vessel. If you look at the content of the lumen. Oh, wait, no, I'm sorry. No, because here we can see red blood cells, which excludes the possibility that this is lymphatic vessel. But definitely there are lymphatic vessels. We cannot say what kind of nerves are because they look the same. So, but branches, yeah. So that's it. Not extremely complicated. Any question? If not, then go on. We continue with the ovary, and we have two ovary sections, 72, 72, underline 2. Let's open both. We will use both. So, ovary is an analog organ to the testis, which is in the middle. And they are considered analog organs because both have the same two major functions. So what are the major functions of these organs? To produce matured gametes, and the other one, hormone production, sex hormone production. So in case of ovary, these two sex hormones, which are worth mentioning, is estrogen and progesterone. Okay, so let's start with this 72 underline 2. Well, the ovary is an extraperitoneal organ, which means that it is connected to the broad ligament, but it doesn't cover the entire surface of the ovary. And this territory is where the broad ligament is connected. This territory is called mesovary. Mesovary, one word. So on this side and on the other side here, there should be some peritoneal coverage, which ends somewhere. It's difficult to tell where it ends over the surface, but somewhere ends because not the entire surface is covered by peritoneum. So that's why it is an extraperitoneal organ. And there is an author's name for that closed line which separates the of the ovary which is covered by peritoneum and which is not covered by peritoneum. How do we call this closed line of separation of the two territories? Line of Fabry, yes, line of Fabry. Well, you know, it is difficult to spot where this line of Fabry may be. Well, actually, this line of Fabry is represented by two dots, actually, on the two sides of the section. It's difficult to spot because, you know, there is a tendency to lose the mesothelial layer from the surface. So, if we can see mesothelium, then we can say it is still peritoneal covered territory, but if we cannot see, it doesn't necessarily mean that at that territory there was no peritoneum before. It is still possible that we just lost it from there. So, if we enlarge, let's see what we can see. Well, for example, here I can see some squamous cells on the surface, which indicates that at least at this territory there was peritoneal coverage. Well, maybe here also you can see that the peritoneum artificially detached here, but afterwards, after a while, this is the squamous here, but you know, if we go far away, let's say here, here on the surface I can see mostly cuboidal cells, okay? Which means that this is probably a territory which was not covered by peritoneum. And this simple cuboidal cell row here is called germinal epithelium. This is a name given in the past, and it is a wrong name because it indicates that wrong idea that these cells give rise to the germ cells in the ovary. This was the belief in the past, and that's why people call this cell row as a germinal epithelium, which still is used, but it is the wrong name. So underneath, we can see a dense connective tissue territory, which is rich in collagen fibers. This is called tunica albuginea. Tunica albuginea. And below the tunica albuginea, the parenchyma starts, and the parenchyma is divided into a peripheral cortex, and the central medulla. This medulla is a loose connective tissue rich in nerves and blood vessels, and these nerves and blood vessels enter the ovary through this territory, through the mesovary, may also be called hilus. Regarding the cortex, the cells here, called stromal cells, these have elongated nuclei, and they usually arranged and described as giving a characteristic pattern, just like how water is going down through the sink, so in a spiraling manner, and this is reflected in the name. This tissue is also called spinocellular tissue, spinocellular connective tissue, due to this kind of rotating arrangement of cells. Major structures in the cortex are the ovarian follicles. Ovarian follicles are here and at different stages of the development. Well, the ovary, just like the testis, is derived from the primitive gonads. Well, most of it, the stroma, is given by the primitive gonads, which is from the intermediate mesoderm. And at the beginning, this called primitive gonad because look the same in the two sexes at the start of the intrauterine life, and it will differentiate towards being ovary or testis after arrival of the primordial germ cells, which are born originally where primordial germ cells appear first, where no, they migrate to the yolk sac, but from there the epiblast, they are born originally in the epiblast. First semester embryology. Everybody paused. So from there they migrate to the wall of the yolk sac, and from the wall of the yolk sac through the dorsal mesentery they migrate to the primitive gonads. Without arrival of these primordial germ cells to the primitive gonads, gonad development stacks. So that's colony of cells need to arrive at the primitive gonad, because otherwise no further development of the gonad. So these primordial germ cells divide mitotically and give rise to oogonia during intrauterine life. And then oogonia divides further mitotically and give rise to what? Primary oocytes. And by the time the baby girl is born, in her ovaries there are already primary oocytes. In which state? In the prophase of the first meiotic division. And it means that girls already more developed in this respect when they are born relative to boys, because in boys there is no primary spermatocyte at this time. Primary spermatocytes appear, which are the analog to primary oocytes. So primary spermatocytes appear only at the start of puberty. Okay, now so what is an ovarian follicle? An ovarian follicle is actually a structure which contains one oocyte plus surrounding follicular cells. And there are different kinds of ovarian follicles. The most primitive ones are called primordial follicles. Primordial follicles. And I show how these primordial follicles look like this. So this is the oocyte. And this is the nucleus of the oocyte, which is euchromatic. And this is the cytoplasm of the oocyte. And at the outer surface of this primary oocyte, there are squamous cells. Squamous cells. These are the follicular cells. And they surround in a single row the oocyte. So this is the structure of the primordial follicle. When the baby girl is born, in her ovaries there are just primordial follicles, no more developed follicle as yet. And nothing really happens to these primordial follicles until the start of puberty. When the puberty starts, the first ovarian cycle starts under hormonal changes. And several, something like 12 or 20 of these primordial follicles somehow are selected, we don't know exactly how, and start further maturation in each ovarian cycle. This happens. And this further maturation means maturation of the follicular cells and also maturation of the oocyte. In this first half of the ovarian cycle, which is called follicular phase because it represents maturation of follicular epithelium, it is under the control of the FSH, follicle stimulating hormone, which is produced by what? By the anterior lobe of the pituitary gland. Remember that when these primordial follicles start to mature further, the oocyte is still a primary oocyte, which means it already started its first meiotic division before the girl was born. And after tens of years, this first meiotic division will be completed during the first half of the cycle. Which means, because a further period of the women is until 45 or 50, something like that. So between the start of the first meiosis and completion of the first meiosis, the time delay may be decades, maybe even 4 decades. And that creates one biological problem, because in this unstable state, so division already started but hasn't completed, this is a vulnerable state for the cell. As the mother is aging, genetic problems are getting more likely. This is one reason why this the probability of mutation is increasing with the age of the mother, because in oocytes are in this vulnerable stage for a longer and longer period before they might be fertilized. So, yeah. Okay, so let's assume that this primordial follicle is somehow selected for further maturation in one of the ovarian cycle. What happens? This follicular epithelium, instead of being simple squamous epithelium, will be a simple cuboidal epithelium. So follicular epithelium will be higher. So this is a transition. So this is still I would say, a primordial follicle, because these follicular cells here are still squamous, but not so very flattened as before. Also, I would like to call your attention that this is the cytoplasm of the oocyte, but the nucleus is not in the plane of sectioning, so that's a complication. So this is a transition between primordial and primary follicle, because look, this is getting cuboidal now, this follicular cell. This is even more cuboidal, as you can see. Again, the oocyte nucleus is not in the plane of sectioning. Okay, look at this. This is rather good example for primary follicle. So in primary follicle, there is a single row of cuboidal cells, as follicular cells. And you know, here, outside the follicular epithelium, we have the basement membrane. This was visible already in the primordial follicle as well, because these epithelioid cells, the follicular cells are epithelioid cells, are separated from the connective tissue of the stroma by the basement membrane. Okay? Later, if this follicle matures even further, then this simple cuboidal epithelium becomes stratified cuboidal. I'm not sure if I can illustrate this, but let's have a look. This is not. I think there is no good example for this in the section, but I hope you can imagine. So the secondary follicle contains a stratified cuboidal follicular epithelium. Now, so far we talked about, okay, I need to add that as the follicle is maturing, further maturation may be arrested at any time, at any stages, and after that the follicle will go atrophic, meaning that it will degenerate. This can happen at any stage of development, okay? And because less and less follicle reaches more and more developed stages, usually there is just one of the maturing follicle will reach full maturity by the time of ovulation. Sometimes more, but usually just one. And the final stage of follicular maturation is when vacuoles appear among the follicular cells, and later these vacuoles, these little cavities, will unite and form a single big cavity, which is called antrum. So here, all these very big structures are actually tertiary follicles, and inside this lumen is the antrum, which is filled up by a fluid called liquor folliculi. Liquor folliculi. Where? Here? No, I would say no. At least I'm absolutely not sure about this. It looks like that, but you know, if it was a secondary follicle, we should assume that still there is no antrum, which means that inside this something should be the cytoplasm of the oocyte. So the oocyte should be as big as this, which is, I think, impossible. It's too big to be a single cell. okay? Because for comparison, this is the oocyte. It can be a bit bigger because this is still a primordial follicle, but it cannot go this big. So this is just a tertiary follicle, like all these, but in more tangential view. That's why the lumen is not so big. Do you understand the reasoning? Okay. If you look at the handout, there is a secondary follicle illustrated there, but as far as I know, it was borrowed from a book to be able to illustrate it for you. It is not from all sections. Okay? If you look at the coloration is a little bit different relative to the other ones, which was a sign of having a different source. Okay. Now, here these tertiary follicles are not completely seen. We need to look at the other ovary section, the 72, which contains also tertiary follicles, but one of them, this one, is very luckily sectioned, meaning that this part, which is called cumulus ophorus, this evaginating part, is in the plane of section. So here you can see the full structure of a fully matured tertiary follicle, also called Graafian follicle. So a fully matured tertiary follicle is the Graafian follicle with capital G. So from that point on, when the antrum appears, we call the follicle as tertiary follicle. And remember, can you remember the previously seen follicle, what you proposed to be a secondary one, and I didn't accept it. The inside looked exactly this, which also proves that it was something like this. So the cavity, the antrum of a tertiary follicle. Okay, so it is not a secondary follicle. So here we have a fluid, lymphofolliculate, full of prostaglandins, glycoproteins, and water, because protein-like materials, that's why the eosinophilic appearance. And the follicular cells now got stratified very much, and this follicular cell population is called now as granulosa cell population, or membrana granulosa. Well, it's not a good name, and it's not a membrane, but it is called a membrana granulosa or granulosa cell population here. These are the derivatives of the follicular cells. In one territory, this granulosa cell population is more numerous, and this cell population is evaginating towards the antrum, and this territory is called cumulus ophorus, and in this territory we have the oocyte. Again, the nucleus is not visible, but the zona pellucida, this eosinophilic rim, is visible, which is a glycoprotein coat around the oocyte. And outer to the zona pellucida, in the immediate vicinity, the granulosa cells, which immediately surround the zona pellucida, are called corona radiata cells. Corona radiata cells. Now, regarding the wall, after the granulosa cell population, or membrana granulosa, we have theca interna. Theca interna. This is derived from stroma cells. And theca externa, which is also derived from stroma cells. theca interna and externa, and between theca interna and externa, characteristically there is a rich capillary meshwork, as you can see here, which consists of capillaries with fenestrated endothelial lining. And that's important because these capillaries receive the hormones which are created here. In the first half of the cycle, mostly estrogen is created by the cells in this hole. In the second half of the cycle, mostly progesterone is created, but some estrogen as well. So these hormones are created by the granulosa cells and the theca cells. Of course, there is no firm, clear boundary between theca externa and the rest of the stroma, but you can see that it is somewhere here, this external border of the theca cell population. So these theca cells and also the granulosa cells undergo a kind of transformation to make these cells real hormone producing cells. So because the stromal cells are originally connective tissue cells, we can say that the stromal cells, well, some stromal cells, which will later on give rise the theca interna and externa, these stromal cells will undergo an epithelioid transformation. So connective tissue cells will become similar to epithelial cells in that they can be able to synthesize and release certain substances in large amount of hormones. Okay. Now look at this. This is a better example. So this is a follicle, which I think is a kind of transition between primary and secondary follicle. Because in certain territories we have a simple cuboidal follicular epithelium, but here it looks like already stratified. Okay? So that's the transition between primary and secondary follicle. Outer to the follicular epithelium, we have the basement membrane, and around the oocyte, the sign of the zona pellucida is visible. And so far we concentrated on maturation of the follicular cells, but also the oocytes mature. Because during the first half of the cycle, the first meiotic division will be completed, and immediately the second meiotic division will start. But the second meiotic division will also be arrested, but in this case in the metaphase. So in the fully matured tertiary follicle here, the Graafian follicle, the oocyte is already secondary oocyte, because already completed its first meiotic division and started the second one, but it is arrested in the metaphase. Now the second meiotic division will only be completed if fertilization occurs, otherwise it will never be completed. Now we need to talk about ovulation, which happens in the mid-time of the cycle under hormonal effects. There is a FSH and LH surge, which is correlating with the time of ovulation. So what happens during ovulation? In short, what can you say? What happens during ovulation? The oocyte leaves the ovary, but only the oocyte. What else?
Tertiary follicles have to rupture. Tertiary follicles have to rupture.
Obviously, for this, it is needed that the wall of the tertiary follicle ruptures, yes. But what structures will, and also the wall of the ovary ruptures, and the oocyte leaves the ovary, but what else is leaving the ovary during ovulation? The zona pellucida and the corona radiata cells. All these structures leave the ovary during ovulation. And in a good case, usually, normally, what is ovulated gets into what? In the uterine tube. The uterine tube, also called oviduct, also called fallopian tube. It's a paired structure which has an opening which opens freely to the abdominal cavity, and the other end is connected to the uterus. And I think I have a figure somewhere about this. Yeah. This is from a Hungarian book, but never mind. So this is the vagina. Here is the uterus, obviously connected to the vagina. And one of the ovaries, the second ovary, it's a paired organ, and the uterine tube here and here also paired. And as you can see, one end of the uterine tube is freely ending in the abdominal cavity in the vicinity of the ovary. And the other end of the uterine tube is connected to the cavity of the uterus. And just before ovulation, we don't know exactly how, obviously it's kind of chemotaxis, this fimbriated end, you can see these kind of elongated fringe-like structures at the end of the uterine tube. These are called fimbriae. These get in close contact with the surface of the ovary, exactly there where ovulation will happen. It's a mystery, but this is happening, and these fimbriae can move and can sweep the oocyte into the lumen of the uterine tube, in most cases. However, sometimes it is missing, it fails, this mechanism, and the oocyte will not go into the uterine tube, but remains in the abdominal cavity. And that creates a danger, because if sperm cells are deposited into the vagina, they travel through the vagina, through the uterus, through the uterine tube, and they may leave the uterine tube and enter the abdominal cavity and find the oocyte somewhere in the abdominal cavity, anywhere, and may get fertilized. So it means that an unusual site of embedding will face an unusual site in the abdominal cavity will face the process of embedding, which is not prepared for this, and that may lead to so-called ectopic pregnancy, which is meaning that the pregnancy is outside the uterus, so it's an unusual location. And you know, this is dangerous for the mother and also for the implant, because it may lead to severe internal bleeding in the abdominal cavity. Okay, but usually the oocyte goes into the uterine tube. Now, So what remains inside the ovary after ovulation? Well, the granulosa cell population and the theca cell populations remain here. Okay? And the granulosa cell population and theca internal cell population will undergo luteinization, which means these cells will be affected by the LH, by the luteinizing hormone, a product of the anterior pituitary gland. And the epithelioid transformation will happen, and these cells will become very much hormone producing cells, produce lots of progesterone and some amount of estrogen during the second half of the ovarian cycle. Because in the first half mostly estrogen is produced by the follicular epithelial cells, and in the second half granulosa cells and theca cells will produce mostly progesterone. The first half of the cycle is called estrogen phase, the second half is called progesterone phase, referring to the dominant hormone which is produced inside the ovary. And these granulosa cells and the theca internal cells will become granulosa lutein cells and theca lutein cells. They have LH receptors, so LH hormone will affect these cells. That's why they become hormone producing cells. And in the third ovary section, we will see the so-called corpus luteum, which means in Latin that yellow body, because this structure, which is formed from the wall of the Graafian follicle, it will form a yellowish body inside the ovary, and that yellowish body in Latin is called corpus luteum. So this is an endocrine organ actually, which develops inside the ovary from the wall of the Graafian follicle in the second half of the cycle under the effect of the LH hormone. Okay. So let's look at the corpus luteum. Yes?
Excuse me, Professor, can you show one more time the difference between the cumulus orphus and corona radiata?
So cumulus orphus is this whole invagination of granulosa cells which contain the oocyte and the zona pellucida. So this whole something here is the cumulus orphus. Corona radiata cells is a subpopulation of granulosa cells, those cells which immediately surround the zona pellucida, these ones. Alright? Well, you need to refer to embryology here because we learned in the first semester that zona pellucida is a glycoprotein code, contains zona proteins, and zona protein 1, 2, and 3, and zona protein 3 acts as a species-specific receptor for sperm cells, and when the sperm cell attaches to it, it initiates certain changes. What kind of reaction occurs inside the oocyte? Not immune reaction. Everybody passed embryology somehow. two processes, both are called reaction. So two kinds of reactions occurs when the sperm cell is connected to the ZP3 receptors. What? Okay, what reactions occurs at this time? No. Acrosome reaction also occurs, but it happens before, because acrosome reaction is the reaction during which the content of the acrosome from the head of the sperm cell is released, which is nothing else than enzymes, whereby the sperm can disrupt the corona radiata, the zona pellucida, and the plasma membrane of the oocytes, which all have to be penetrated through in order to be able to fertilize. So that's the acrosome reaction. What happens next? Two other reactions. Cortical reactions and zonal reactions. So what is cortical reaction? What goes from where to where? Cortical granules which are in the oocyte, which are actually vesicles which contain enzymes. They are released, and how they will act. Now these enzymes will inactivate the ZP3 receptor, and that's that inactivation what we call zonal reaction. Ooh. This is the ABC of the title called fertilization. I don't want to predict anything, but during the final exam, if you don't know these, that creates a problem. Okay, now let's forget about this for now. And I just would like to open which one? What was the film? 73.
Yeah.
So remember that during the oral exam, this semester and also next semester, after next semester, the final exam, we can ask embryology questions as well, at any time. And those may be different from the published questions. So, this shows the ovary in the second half of the cycle, and this big structure, this big structure inside, this is the corpus luteum, inside the ovary. Okay? So, remember what happened. So, ovulation occurred. Obviously, the liquor folliculi, the fluid content of the antrum, lost. Because the wall of the Graafian follicle ruptured. And it includes some bleeding, because the blood pressure, which previously kept the balance with the hydrostatic pressure of the liquor folliculi, this balance was disrupted because the liquor folliculi was lost. Blood pressure cannot accommodate immediately, so relatively speaking, the blood pressure becomes higher, and it will lead to rupture of small capillaries in the wall, so some bleeding occurs, obviously. Anyway, and that blood is flowing into the cavity of the late Graafian follicle. Now this structure, which consists of granulosa cells, theca interna and externa cells, and inside the late antrum there is some blood. This structure is called corpus haemorrhagicum. corpus hemorrhagicum. So this pinkish territory here, inside the corpus luteum, is the territory of the late antrum of the Graafian follicle, which was filled up by blood. And after filling up by blood here, this blood clot is degenerating, and macrophages appear inside. Can you see these cells inside this blood clot, this degenerating blood clot? There are around these cells with eosinophilic cytoplasm, macrophages. They try to clear this area from this remnant of blood. Okay. In the meantime, granulosa cells, under the effect of the LH hormone, undergo so-called luteinization, which means they become hormone-producing cells, these ones. And now they are called granulosa lutein cells. Granulosa lutein cells. They are cells with basophilic cytoplasm. Granulosa lutein cells. Now, theca interna cells outside to the granulosa lutein cells become theca lutein cells. Theca lutein cells, these are smaller cells relative to the granulosa lutein cells, theca lutein cells. So this cell population is the theca lutein cell population. This is derived from the theca interna cells. And these two cell populations together produce lots of progesterone and some estrogen. Now, what is the later fate of the corpus luteum? It depends on whether or not fertilization and implantation occurs. If no fertilization and implantation occurs, then the corpus luteum will start to degenerate towards the end of the second half of the cycle. So the lifetime of the corpus luteum is about 10, 12 days. And that's because the LH level is dropping. And LH is needed to maintain the corpus luteum, because these granulosa lutein and theca lutein cells have LH receptors to have stimulus. And if no more LH, corpus luteum will start to degenerate, will shrink. In the meantime, these macrophages, phagocytose, the remnants of blood clot, they get disintegrated, and they release a fibroblast growth factor, a substance which initiate proliferation and growth of fibroblasts around. And these fibroblasts will create lots of molecules for connective tissue fibers. So this area where this degenerating blood clot is now, which is at the territory of the late antrum, this territory will turn to a fibrous connective tissue because lots of fibroblasts will appear here later. And that is the so-called corpus fibrosum. Corpus fibrosum, the next stage of degeneration. Corpus fibrosum. In the other section here, well, I don't know, maybe this one or the other one, I'm not sure. There is something like that. Yeah, so look at this structure here. This is the late corpus luteum, and this is the territory where the blood clot was. And if you enlarge here, fibroblast cells already appeared. and produce lots of collagen fiber. You can already see the fibers. Okay? So this is something like corpus fibrosum, or you can say this is already the start of something which we call as corpus albicans. That's the last stage of degeneration. The corpus albicans means whitish body, because this is whitish to the naked eye without artificial coloration. But this is some kind of transition between corpus albicans and corpus fibrosum. And a very characteristic corpus albicans is seen in the other section, in the 72. I show you. So after the stage we just could see, the next stage of degeneration is the real corpus albicans, which is again quite characteristic. Look at this structure here. So it is much smaller, and it has a kind of spiraling appearance. Okay, so this is a typical corpus albicans, the remnant of the late corpus luteum. And this is another example, corpus albicans. And yeah, this is also an atrophic follicle. It's a kind of degenerating connective tissue fibers are seen here. So after a while, the remnant of corpus luteum will be indistinguishable from the rest of the stroma, because the tissue will get so much degenerated and so much cleared by macrophages that actually it will disappear, but it takes a long time. Okay. Now, we talked about the fate of the corpus luteum in case of no pregnancy. But if there is pregnancy, the fate of the corpus luteum is different, because it will not degenerate after 2 weeks, but will persist at least for 3 or 4 months during the first trimester, at least during the pregnancy. Why is that? How the corpus luteum knows that pregnancy occurred and it shouldn't degenerate yet? What is the message? What hormone? No. Human chorionic gonadotropin, HCG, is the hormone which is messaging. Human chorionic gonadotropin, which is produced where? By? Not by the embryo, well, no. Basic embryology again. I am a little bit nervous for your next exam in embryology. It is produced by the syncytial trophoblast of the developing placenta. Syncytial trophoblast, which is part of the blastocyst, you know. And it imitates the effect of LH. That's why corpus luteum will not degenerate so early. And that's important because to maintain pregnancy, lots of progesterone is needed. And without enough progesterone, there is a spontaneous abortion. And progesterone is produced by the corpus luteum. Later, enough progesterone will be produced by the placenta itself, but before the placenta reaches that stage of development to be able to produce enough progesterone, until that stage, corpus luteum needs to continuously produce progesterone. And this is achieved by HCG, which is also a basis of an early pregnancy test because HCG also appears in the urine. A 10-day-old pregnancy can already be detected by detecting HCG level within the urine of the mother. So that's the basis of the early pregnancy test. So that's why also very important to know this, and I'm slightly disappointed that you don't. All right. So we have one more section, which is the mammary gland. 81. And this is actually the mammary gland in the lactating stage, which means in the stage when it really produces milk. So it is after delivery. Otherwise, it is a non-lactating gland. Lactation, meaning milk production, starts right after a baby is born, under hormonal effects. So how can you see that this is a lactating mammary gland? You can see here by comparing the amount of secretory units, which are these bluish territories, comparing the territory of these secretory units with the amount of white adipose tissue, which is the yellow. Here, the majority is given by secretory units. That's why we can say that this is a lactating gland, lactating mammary gland. Otherwise, if adipose tissue would be the dominant, then we should conclude it is a non-lactating mammary gland. And this lactation, so development of secretory acini and secretory cells and initiation of milk production is due to hormonal effect, due to which hormone, which shoots up after delivery, which hormone facilitates milk production in the gland. Prolactin. Prolactin, which is produced by the anterior lobe of the pituitary gland. Yes. If you see this organ, you can define kind of units surrounded by connective tissue septae, like this. These are the lobules. So this is a lobulated organ. Within a lobule, we have obviously secretory units and intralobular ducts, which are difficult to distinguish. It is not a requirement from you. But inside secretory units, there are two types of cells, which are also difficult to distinguish a little bit. Because one kind of cell produces the lipid components of the milk. These tend to be having pale cytoplasm because the lipid was dissolved. These lipid producing cells, so one major chemical in the milk is lipid. And there are other cells whose cytoplasm is darker. Those produce the proteins, mainly. The other major chemical in milk is the protein. And the interesting thing is that the lipid-producing cells use apocrine secretory mechanism, and the protein-producing cells use merocrine secretory mechanism. Lipid-producing cells, apocrine. Protein-producing cells, merocrine. don't expect me not to ask what is merocrine and apocrine secretion in this context during the exam. Don't expect that. I will. Okay? And what is this red line around the secretory unit? Basement membrane. And what is this cell? This is a myoepithelial cell, just like in salivary glands. Cell with processes around the secretory cell, and this cell can contract and squeeze the secretum out of the secretory cell. Also, you can see among the secretory units cells like these. What do you think about these cells? They have round nucleus, basophilic cytoplasm, and the nucleus is not central. What cell is this? Oval-shaped cell. Don't disappoint me so much today. Plasma cell, somebody said. Plasma cell, which produces what? Antibody. Which kind of antibody is produced mostly here? No. No B, I it doesn't exist. Maybe you will discover later. A. And A type is very unique because it is called secretory immunoglobulin, because it is the immunoglobulin is released by this plasma cell, and the acinar cells take them up at the basal aspect because they have receptors over the basal plasma membrane. So immunoglobulin A will bind to the receptor on the surface of the acinar cell, the secretory cell, and receptor-mediated endocytosis will occur. Then transcytosis will occur, so together with the receptor, the IgA will be transported through the cell and released over the apical membrane, released into the lumen, so it will become part of the milk to give passive immunity for the fetus, or for the newborn. And this IgA takes part of the receptor with it. That's why this was secretory immunoglobulin. This secretory component gives kind of protection to this immunoglobulin against degradation. Okay. And maybe one more thing, or two things. This one is a duct in the interlobular connective tissue septum. So this is an interlobular duct, and you can see blood cells even inside the milk. This is not a vessel because this is not an endothelial lining here. Okay, cannot be a vessel, but still there are blood cells here, because in early milk called colostrum, starting with C, colostrum, the early milk or pre-milk, still contains blood cells, surprisingly. So this is still colostrum, so it's this sample was taken shortly after delivery. That's the conclusion. And another thing what I just would like to show, this is another interlobular duct, by the way. And another thing that you can see, look at this, lymphatic vessel. Lymphatic vessel here. So, that's it. Of course, like adipose tissue, so half of the histology can be asked in relation to this section. Okay, glandular epithelium, blood vessels, secretory mechanisms, immunology. What do you want? So it can be an easy section if you know everything what you already should know. But if you don't, it can be a very challenging section. Any question? If not, then thank you very much. Please turn the computers off before you leave.